Hello, everyone, and welcome to today's live broadcast, Exosome miRNA sequencing for biomarker profiling using the ION S5 system and ION Total RNA-seq kit, presented by Dr. Lesley Cheng, postdoctoral researcher. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar, presented by LabRoots and sponsored by Thermo Fisher Scientific. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, we will be conducting a few audience polls. The poll will pop up on your screen, and we appreciate your participation. Our first poll of the day: which RNA application would you like to learn more about? Select all that apply. Whole transcriptome RNA sequencing, targeted RNA sequencing for transcriptome analysis, targeted RNA sequencing for selected gene expression analysis, small RNA sequencing, non-coding RNA sequencing, fusion gene detection, or none of the above. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located in the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button at the bottom left corner and follow the process to obtain your credits. I'd now like to introduce our presenter. Dr. Lesley Cheng is a postdoctoral researcher. She was awarded a Bachelor of Medical Science with honors and a PhD from Monash University, Australia. She is currently based at La Trobe Institute for Molecular Science, La Trobe University, Australia. Her research expertise combines basic and clinical science through the use of novel cell and molecular biology approaches to identify biomarker candidates for diagnostic testing. Currently, her major focus is to develop a minimally invasive blood test for the early detection and monitoring of neurodegenerative diseases such as Alzheimer's disease, Parkinson's disease, and Prion disease. Her research involves detecting disease signatures of RNA molecules called microRNA, which can be collected from small biological vesicles called exosomes that travel in the bloodstream. She is also investigating the functional role of exosomes in neurodegenerative diseases, as they have been found to cause the spread of disease in the body. I would like to turn the presentation over to Dr. Cheng.
Hi everyone, thanks for joining us today on this webinar. I'm assuming that if you are here on today, that you're interested in performing small RNA sequencing on the Ion Torrent platform, and perhaps you will also be profiling exosomes. So a lot of my research does focus on exosomes. They are an extracellular membrane vesicle of endosomal origin. As you can see from this EM image, they are about 50 to 100 nanometers in diameter. They carry nucleic acids, proteins, and lipids. You can see from this bioanalyzer profile that in lane 2, exosomes contain an enriched population of microRNA, tRNA, and small RNAs. And this is quite different to what we see in cells, which is in lane 1 of the bioanalyzer profile. They also contain protein, and as you can see in this lane, that the exosomes protein laddering is quite different to whole cell lysate. One of the RNA species that I am mostly interested in my research is microRNA, and they are a non-coding small RNA species packaged into exosomes. So microRNA are transcribed in the nucleus as primary microRNA of several thousand nucleotides long. It is then further processed by an RNase 3 type enzyme, Trobe, to precursor microRNA before being transported from the nucleus to the cytosol via exportin 1. From here in the cytosol, it's further cleaved by dicer to produce a microRNA duplex of about 22 nucleotides long, where one strand is then incorporated into the RNA-induced silencing complex together with ARGO2. From here, microRNA can then be bound to the target messenger RNA and inhibit translation of the protein. From here, microRNA can also be bound to RNA binding protein and be secreted into the bloodstream. They can also be packaged into exosomes and then be secreted into the bloodstream. From here in the bloodstream, exosomes can then be taken up by a recipient cell, where its microRNA contents can influence translation in the recipient cell. However, as we know, we can collect blood and then isolate exosomes from the blood. And so now exosomes have been used as a major source of biomarkers and potentially contain diagnostic indicators for various diseases such as cancer, infectious diseases, and brain diseases. However, when you collect blood, microRNA can be found in various components of the blood, such as in the neat serum or plasma, or in the cellular component, which contains red blood cells or white blood cells, etc. So when profiling for microRNA in blood, it would be important to find out where the strongest disease signature lies. In terms of the cell-free plasma, you can find microRNA bound to RNA binding proteins or contained in exosomes. In the cellular component, they can also originate from the red blood cells or white blood cells. So some of my work back in 2014 involved trying to understand where the majority of microRNA may be useful when it comes to diagnostic testing or biomarker discovery. So what I did was collect whole blood and isolated for intracellular RNA using PAX-gene tubes. I also fractionated the whole blood to collect for cell-free serum and plasma, where with the cell-free serum and plasma, I performed a sequential ultracentrifugation to isolate exosomes. In order to isolate then the RNA from the exosome palette, I used the miRNA Easy Kit. I compared these ultracentrifuge exosomal RNA profiles with a commercial kit from Norgine Biotech, as well as comparing these profiles to cell-free or neat plasma or serum. This was all collected from healthy volunteers. So running these samples on the bioanalyzer using a small RNA chip, you can see in the PAX genes firstly that PAX gene RNA shows tRNA and 5S RNA as well as microRNA. Not surprisingly, when you treat the PAX gene RNA with RNase A, this is all degraded. When you look at cell-free plasma and serum, you can see that cell-free plasma and serum contains mostly microRNA and slightly the presence of other small RNA. Not surprisingly, again, when you do an RNase A treatment, all the RNA in plasma or serum is also degraded. So what happens when we use exosome samples? Looking at the exosomal RNA that were isolated by the ultracentrifuge, you can see with plasma and serum, they contain the majority of microRNA and other small RNA. And when you treat these samples with RNAs, you can see that the RNA is no longer degraded and potentially protected by the lipid membrane of exosomes. Looking at the commercial kit by Norgine Biotech, the same profile is quite similar in serum, and it's also quite similar in plasma. However, in this bioanalyzer trace, I've shown an example of what happens when there is intracellular contamination, and upon RNase A treatment, you can see the degradation that's happening there. So overall, exosomes contain an enriched population of small RNA, in particular microRNA, that is protected from RNase treatment. And in the bloodstream, there is a high volume of RNases floating in the bloodstream, making exosomes ideal for biomarker discovery. So upon performing small RNA sequencing on these samples, you can see on the y-axis where I've graphed the percentage of matched reads looking for microRNA, smallRNA, ribosomal RNA, and other non-coding RNA, that exosomes contain the highest percentage of microRNA from 31% to 48%. This is in comparison to when you look at neat serum or plasma, where you get about 13 to 14% microRNA, and patched gene tubes 28% microRNA. So essentially, by isolating exosomes, you could potentially improve signal-to-noise ratio for diagnostic testing or biomarker discovery. So the workflow that I use is outlined in this slide. Firstly, I perform sample collection. Today I will be mostly focusing on plasma and serum. However, of course, you can collect exosomes from conditioned cell culture supernascent, and I'll show you a bit later that we've also been able to isolate exosomes from intact tissues such as the brain. And we then go and perform exosome isolation if that is what you are interested in, profiling exosomes. Or you can extract RNA after this, followed by an RNA quality assessment, and then construction of the small RNA libraries, followed by ion torrent sequencing, which takes about 2.5 hours for a run. This is followed then by QC using various QC programs for your NGS analysis, and then followed by bioinformatics analysis. So the ion torrent S5 chip that I mostly use is the 540 chip, where you're able to obtain 60 to 80 million reads. Most of my libraries are between 22 to 200 nucleotides in length, and this mostly comprises of the small RNA family. Members of the small RNA family, of course, there are non-coding RNA as opposed to coding RNA. So the small RNA family consists of smallRNA, pwRNA, microRNA, which are 18 to 25 nucleotides in length, siRNA, and small nucleolar RNA. All of these small RNAs play a crucial role in the regulation of genes, and they control a diverse number of cellular and biological pathways. Currently, there's about 2,500 known microRNA species in the human, and approximately the same in the mice as well. So our lab has really been in the forefront of the EV field, from characterizing exosomes and then followed by being able to profile exosomes. And this really all began in 2007 and 8, where we're able to characterize exosomes isolated from CSF, as well as exosomes from various cell lines, such as those that are infected with the Prion protein, and also our Alzheimer's disease cell models, where we identified the presence of the Prion protein and fragments of APP in exosomes. In 2012, we were able to show the first cryo-EM of Prion-infected cells, as well as being able to sequence microRNA from these Prion-infected cells using back then the solid platform. In 2013, we found that exosome-like vesicles were responsible for cell-to-cell communication in malaria-infected red blood cells. In 2014, our first papers using the ion torrent systems were published, where I was able to profile small RNA in an extensive and comprehensive way from exosomes isolated from various biological fluids such as urine, plasma, and serum. In 2015, I published a paper where I applied this workflow to serum samples collected from Alzheimer's disease patients, where I was able to identify a panel of microRNA biomarkers associated with Alzheimer's disease, as well as melanoma. In 2006, we started publishing on how we believe the prion protein is packaged into exosomes. And in 2007, this year, we've also published a number of papers using the INS5. So our lab has also been part of many tools in development that can be accessed online, such as databases that curate a database of what proteins and microRNA or RNA species are found in exosomes, such as Vesiclepedia, EVpedia, and EVTrack, and they're all online as well. So in terms of exosome isolation, we typically use sequential ultracentrifugation, which is really a universal tool. So in this slide, I've demonstrated conditioned medium, but you can also obviously use plasma or serum, where we perform a 2000 G spin to pellet cell debris. We then transfer the supernatant to an ultracentrifuge, where we then spin at 10,000 Gs to pellet large multivesicles. The supernatant is then transferred again to be spun at 100,000 Gs to pellet exosomes, further by a PBS wash to wash the pellet of exosomes and to be spun at 100,000 Gs. So at the end, you get a pellet of exosomes, and you can characterize the exosomes via Western blotting, looking at exosomal markers such as TSG101, flotulin, and non-exosomal markers such as BCL2, which represents apoptotic bodies, Golgi, GM130, and nucleoporin, which is nuclei. Typically, we also view our exosome preparation by electron microscopy, and using this sequential ultracentrifugation protocol, it usually consists of a crude isolation where sometimes you get other vesicle sizes and sometimes some protein contamination as well. So you can then take this exosome pellet and perform a Optiprep velocity gradient to further purify and fractionate your exosomes. Using a 10 to 30% Optiprep gradient, you float your exosome pellets on top of this gradient, where you can then, upon a 250,000 G spin, collect fractions and then perform a Western blot to determine which fraction your exosomes floated at. So in this Western blot, you can see the exosomes are positive for TSG101 and CD63 in fraction 8 and 9. And you can see in the EM that these preparations are a lot more pure. So in terms of biomarker discovery, we wanted to determine whether it was necessary to perform a Optiprep velocity gradient with exosomes. And we sequenced the fraction 8 and 9 from the Optiprep gradient, as well as preparations of exosomes isolated by the crude method, which is just an ultracentrifuge preparation. And we compared these two fractions on either side, fractions 7 and 8, as well as cells. And you can see that fraction 8 and 9, and the ultracentrifuge exosomes clustered together, suggesting that they have similar small RNA profiles. In this Venn diagram, the common microRNA species, the majority of microRNA species are also the same between an ultracentrifugation prep and also fraction 8 and 9, suggesting that it's probably unnecessary to further purify exosomes for biomarker discovery, and that ultracentrifugation is maybe good enough to obtain the same small RNA profiles for biomarker discovery. So what we've also done is transfer this protocol, which is very similar to isolate serum and plasma exosomes. As you can see that they also are positive for flotulin, transferrin. We've also here profiled PRP, as we've seen the prion protein present in exosomes, as well as CD63. These are the exosomes from plasma and serum under EM, and using nanocyte particle counter, they are all sitting at about 100 nanometers in size. What we've also been able to do in our lab is not only isolate exosomes from, say, what we call circulating exosomes, but also from intact brain tissue. So this protocol has been published this year in JEV, the Journal of Extracellular Vesicles, where we've been able to float the exosomes on a triple sucrose cushion after being able to loosely remove the exosomes gently from the intact tissue, perform a series of ultracentrifuge spins, float it on a triple sucrose cushion, and upon characterization, we have found that the exosomes are floating in fraction 2. So for all of the brain-derived exosome work, we are investigating exosomes in fraction 2. Of course, ultracentrifugation can take a while, and we've also explored the use of commercial exosomal RNA kits, such as the Norgine kit, which I spoke about previously, but also the kits from Invitrogen and Thermo, such as the ones that isolate plasma and serum, as well as the miRNA kit. So going through my workflow for NGS, initially, once you've isolated your exosomes or samples, we perform a small RNA quality assessment using the bioanalyzer. And this typically looks like as what you can see on the slide. It's much more easier to make small RNA libraries, obviously, from cells or tissue. And in this case, where you have not only small RNA but also messenger RNA, we perform a RNase 6000 assay. And this is just to look to see whether the RNA is of good quality, and anything greater than a RIN of 7 is usable for NGS. So you can see here that this particular sample from brain tissue has a RIN of 10, which is great. It has a percentage of small RNA as well as messenger RNA and potentially long non-coding RNA. When you then go and look at the small RNA profile, you can see peaks of tRNA, ribosomal RNA. So with these sort of samples, what I do with the small RNA assay is determine what is the percentage of microRNA. And this percentage of microRNA will give you an indication of how successful your small RNA libraries will be. So this particular sample is 22% after enrichment. And even if the small RNA percentage is quite low, it is necessary, I believe, to perform a small RNA enrichment before continuing on to small RNA construction, as ideally you want to remove as much messenger RNA out of the system before attaching your adapters. So another way where your samples can be enriched with small RNA and not having to enrich it in the lab is basically looking at exosome samples. So as I presented before, serum exosomes usually are already biologically enriched with small RNA. So small RNA enrichment is not required. And you can see in this particular profile that there is a lot of microRNA already. So this is quite different. And ideally, we do need a minimum of more than 1 nanogram per microliter of small RNA. This is quite different to, for example, the small RNA profile from exosomes from conditioned media. Sometimes they are not enriched with small RNA and they can also contain tRNA, but this is okay. Usually they don't contain anything larger than 200 nucleotides, so you don't need to perform a small RNA enrichment and then go ahead with your adapter ligation. In terms of when you isolate exosomes and you're not using a commercial kit that goes from exosome straight to RNA, I do isolate the exosomes from ultracentrifugation protocol and then use Trizol LS to extract my RNA, which also works very well. So then when it comes to small RNA library construction, I use the Ion Torrent Ion Total RNA-seq version 2 library kit. This is the workflow of the protocol, which I'll briefly go through my library quality control and assessment. This protocol has also been published in Methods and Protocols if you want to have a look. This basically involves some of my tricks and also several changes to the protocol to be able to make small RNA libraries from small yields of RNA, in particular from exosomes. So as I mentioned, it's quite straightforward to make small RNA libraries when you have plentiful of RNA, such as in brain tissue or cells. So what we do here is determine the percentage of desired RNA ligated products from the total ligated products. So the barcodes themselves and the adapters, the 5' adapter and the 3' adapter, are usually about 85 base pairs in length alone if you are barcoding your libraries. If there is no insert, you will see that you'll have a peak on your DNA 1000 assay chip at 85 base pairs long. If you have a 10 base pair insert, that means your inserts or your ligated products will be about 95 base pairs. And then if you do have a microRNA insert, your fragments should be sitting at about 105 base pairs long. Anything larger will be about 135 base pairs long and greater. So you can see in my bioanalyzer trace here that I have a number of peaks. And this 109 peak is mostly, could be microRNA and then other small RNA species. So when you perform a small RNA smear test, what I do is gate the entire library, 50 to 300, and then also gate what I'm actually interested in, which is anything beyond 91 base pairs, which will have an insert, and then to 250. And so the fragments that I'm interested in takes up 94% of my library here, which is great. So we basically aim for anything greater than 50% of the desired ligated product. Anything greater than 50 will tend to produce good sequencing run, but obviously the more the better. So the difficult thing is when you start having lower RNA yields to play with, which here we've got 15 nanograms of RNA. And the profile here, you can see a small peak of primer dimers appearing where there is no insert, but overall we have 72% of desired ligated products, which still works in this case. In terms of when you are making small RNA libraries from exosomes, we rarely have heaps of RNA, and in this case we have 1 nanogram of RNA. So what happens with 1 nanogram of RNA? The barcode and the adapters are basically in excess of what the RNA is, of the RNA that is contained in the tube. So upon cleaning your adapters and barcodes, we see that sometimes there is a high percentage of primer dimers. And so this is one of the major issues when we construct small RNA libraries with low RNA yield. And you can see here that the smear test only produced 48% of the desired ligated products. So what I basically do is, in terms of when you're using precious samples, you may have no choice but to accept these primer dimers and filter them out during bioinformatics analysis. Or you can try and site select and clean again. However, I find that you end up losing a lot more than gaining. Of course, you can always try and increase RNA input, but when it comes to using biological samples or post-mortem tissue, obviously those samples are quite precious and you may not have enough to begin with. So what I do with these samples now is basically upon RNA extraction, I then speed evac my entire volume of RNA and use it all. In terms of the overnight adapter, adapter ligation, I perform an overnight incubation, 16 degrees for 16 hours, and I find this greatly improves the success of the small RNA library construction. I also decreased the amount of adapters I put in at the very beginning. And in terms of site selection, using ethanol, I find that exceptional pipetting works very well. Make sure that your pipettes are calibrated and that you are pipetting correctly the volume of ethanol. And what you will get, hopefully, is very clean libraries from 1 nanogram of RNA. So those are my tips of constructing small RNA from low-yield samples. What we've also been managed to do in our lab is also semi-automate small RNA library using the Biomarker 4000 as well. So this has helped a lot with our workflow when we've got many samples. After this, I then, of course, move my samples and libraries to the ion chest. And this instrument has really been great in our lab and being able to give us a lot of reproducibility in terms of our sequencing from here on. In terms of the library concentration on the ion chest, I use 45 picomolar, where I find that this gives us the best sequencing results. To run the ion S5, it's quite simple. What I do is always set up a template run where we have our own template protocol on our system. Here you can see I've called it the Hill Lab Small RNA template, and basically it's quite user-friendly to use, and then you can just queue your runs ready for sequencing the next day. Here is one of our completed runs. So you can see here that this run showed 93% loading on the chip, and the usable reads, we've reached quite close to capacity, which is 79 million reads. You can see the breakdown of each sample that was barcoded on the chip, which reaches about 4 to 5 million reads per sample. In terms of the concentration that I used for template preparation on the ion chest, 45 picomolar, we're sitting at 34% polyclonals, which is usually ideal in terms of template dilution, and we've got 66% clonal, and then all this filtering gives us the 80 million reads ready for bioinformatics analysis. This particular sample, the average read length is about 100 nucleotides long for these samples and small RNA libraries. So this workflow, what I've done is applied this to some of our research involving Alzheimer's disease. Alzheimer's disease is characterized by the buildup of a neurotoxic protein called amyloid beta. And this buildup of this protein causes neuronal cell death and impairment in thinking and memory. The average onset of this disease is 65 years old, as illustrated in this graph. However, we now know that the buildup of amyloid actually occurs 10 years before the onset of symptoms, as you can see here. So if you were diagnosed at 65 years old, the buildup of amyloid may have occurred when you were 55 years old. So treatments aimed at limiting the buildup of amyloid beta really requires methods to diagnose the disease earlier, and so that we can monitor the disease earlier and potentially administer any drugs prior to the buildup of a greater load of amyloid beta. A lot of our work, each participant is PET imaged, and you can see in this figure the buildup of amyloid beta in the brain, as indicated by the fluorescence in this PET image, which is compared to the lack of the buildup of the amyloid tracer in a healthy control brain. So the idea here is that although PET imaging is quite accurate, it's not ideal for routine testing, and our lab has been focused on developing a blood test, which is less invasive, it's more convenient, and it's also cost-effective as well. So we collect our samples from from a cohort in Australia called ABLE, the Australian Imaging Biomarkers and Lifestyle Study of Aging. It was launched in 2006. It's one of the largest longitudinal study of its kind in Australia. It contains currently 1,600 participants, where the minimum age is about 60 years old. We have Alzheimer's disease patients, those with mild cognitive impairment disorder, and also controls. And every participant undergoes a battery of assessment every 18 months, where they come in and perform neuropsychological tests. They get PET imaged, and we also collect their blood as well. So back in 2015, I published a paper where we were able to apply this small RNA workflow through a number of patients with Alzheimer's disease and also with the healthy controls. And I found a panel of 16 microRNAs that were significantly deregulated in the AD. So this comprised of two major clusters, 13 microRNAs which were upregulated and 3 which were downregulated. And using random forest modeling, we were able to show that this panel of microRNA was able to predict for AD with high accuracy as well as sensitivity and specificity. Here are the 16 exosomal microRNA biomarkers. As you can see in the red, that some are upregulated in the ADs and others are downregulated. In terms of sensitivity and specificity, we were able to attain 87% sensitivity as well as 70% specificity. What we've now moved on to do is to see whether those 16 microRNA were also differentially expressed in the brains of post-mortem tissue of AD patients. So here you can see that we've been able to compare post-mortem tissue from control samples, an N of 8, as well as those from AD patients or subjects. We were able to isolate brain-derived exosomes from these tissues, and what I also did was also look at the profiles from the whole brain tissues as well. What I wanted to look at is whether there was any differentially expressed microRNAs in these AD brain-derived exosomes and also in the whole brain, and compare them to the data set that I previously sequenced using the serum exosomes. And what we wanted to do was to see whether there is any common differentially expressed microRNA between the brain as well as the serum in order to potentially develop a liquid biopsy of the brain that represented more closely to what was happening in the brain. Initially, what we wanted to see whether there was any small RNA and microRNA in these brain-derived exosomes as well as whole brain. And you can see this was performed on post-mortem tissue from healthy subjects, where fraction 2, as I previously showed, where the brain-derived exosomes are found. And you can see that 74% of microRNA are found in fraction 2, which is the brain-derived exosomes, whereas in whole tissue, there is 12% microRNA. So from here, we then sequenced the brain-derived exosomes as well as whole brain from the post-mortem tissue of AD subjects as well as healthy controls. And I compared these small RNA profiles to serum exosome profiles that I previously sequenced. So this graph basically allows me to determine what common microRNA species are found in the brain and serum. On the y-axis, I have graphed the fold changes found in whole brain tissue, AD versus healthy control, well, two-fold change, where the bottom of this graph, you can see basically all the microRNA species that are downregulated. And on this side of the graph are those that are all upregulated. In the x-axis, I have graphed the brain-derived exosomes, AD versus healthy control. On this side of the graph are those that are downregulated, and on this side of the graph, they're all the ones that are upregulated. Hence, in this quadrant, these microRNAs, for example, are upregulated in the whole brain, but downregulated in the brain-derived exosomes. So they are of opposite expression. Whereas in this quadrant, they are within the same direction, being upregulated in both sample types. So essentially, in terms of picking up a biomarker in both tissues, ideally using qPCR, the second quadrant here would be ideal to pick up, being upregulated in both tissues. What I also did was graph also the microRNA that were found in serum exosomes versus the brain-derived exosomes to determine whether there was any common microRNA species in the serum where I could pick them up as a diagnostic indicator that is reflective of what is happening in the brain. So it would be this quadrant here. Interestingly, some of these microRNA that were identified in these studies are predicted to hit messenger RNA targets associated with Alzheimer's disease, such as APP processing, apoptotic cell death, as well as ER stress and pathway involving GSK3B, as well as the NMDA receptor. This is using target scan and also KEGG pathway analysis. Part of our lab, we've also been able to develop tools to analyze small RNA, in particular from files generated by the Ion Torrent, which is currently available from this link here. So overall, today hopefully I've shown you that exosomes provide a protective environment for RNA cargo as seen in the first slides, looking at bioanalyzer profiles from exosome samples and treated with RNase-A. Exosomes can be isolated from biological fluids and are enriched with microRNA. Isolation method should cater for high throughput biomarker screening. So it's best to try and optimize your RNA isolation and increase RNA yield before going into small RNA library construction. So tips for improving exosomal small RNA libraries is basically to maximize exosome yields through the various isolation methods you can use to isolate the exosomes, followed by maximizing RNA extraction, exploring various RNA kits, and then followed by concentrating the RNA using a speed evac. If you don't have enough RNA to begin with, you can speed evac it and then use the entire volume for small RNA library construction. Overnight ligation reaction definitely helps a lot, and decreasing the adapter input at the beginning. You can also increase PCR cycles during the PCR amplification. I wouldn't do more than 18-20 cycles though. And then you can compare the small RNA profile with the resultant library profile. So looking at your RNA profile and determining how successful you will be at the very end upon library assessment. In terms of selecting biomarker candidates, so once you've already run your samples through the S5 or the Ion Torrent, you can have a look at what sort of reads you've obtained, and after some experience, you'll see that sample collection does affect your bioinformatics analysis a lot, and so careful sample collection is very important. Experimental design is also very important, and then filtering for abundant species for analysis to remove biological variability. I always check the raw reads between the normalized reads before validation by qPCR. This is to ensure that you are trying to validate the most abundant species that have appeared in your NGS data. And also a great tip is to use your NGS data to find endogenous controls for normalization when it comes to qPCR validation. So this is our lab here, and all the people that are involved in this work are all highlighted here. Our lab is funded by various funding agencies as well as government agencies. So that is it for my presentation, and we will now go to some questions.
Thank you, Dr. Cheng, for that informative presentation. As you said, we will now start the live Q&A portion of our webinar. If you have any questions you would like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on the screen and click the send button. We'll answer as many questions as we have time for. If by chance we run out of time before we have answered your question, Dr. Cheng will follow up via email following the presentation. Okay, let's get started. Our first question is, okay, Dr. Cheng, what are your thoughts on ExoQuick versus UC?
I think the ExoQuick product, when it first came out, we were really unsure on the level of contamination that was produced by the ExoQuick protocol. Since then, I think they've greatly improved. From characterizing the exosomes that are produced from the ExoQuick reagent, we do see several sizes of vesicles. So it's probably a lot more different to ultracentrifugation, but I think if you use ExoQuick, as long as you get reproducible results and potentially looking just for a biomarker and process all your samples by ExoQuick, it's fine. What we did was compare the profiles of ultracentrifugation to commercial kits such as ExoQuick, and then picked the commercial kit that produced the closest profile to ultracentrifuge. Obviously, for biomarker discovery, you want to be able to isolate exosomes as fast as you can. Diagnostic labs really don't have time for ultracentrifuge protocol. So in terms of ExoQuick, the profiles do look a little bit different, but as long as you remain consistent, you can use it for biomarker discovery.
Here's a great one. Have you used enriched small RNA from breast cancer cells?
In terms of the exosomes that are secreted or just the cells? I'm assuming maybe just the cells. We haven't done it on breast cancer cells, but it wouldn't be too different from sequencing small RNA from any other type of cell line. So what I would do is isolate the RNA from your cell line, being in this case breast cancer cells, making sure the RIN looks good, and then performing an RNA enrichment. So these days, when I am making small RNA libraries from cells, I perform a small RNA enrichment no matter what, just to increase the chances of the adapters binding to only small RNA and nothing else.
Here's a question. What does the filter 0.22 micrometer mean?
Sorry, that is the filters that we use to filter our conditioned supernatant to remove any larger vesicles that may be present. So before carrying forward to the ultracentrifugation spins, we filter our conditioned supernatant with a 0.22 micron filter. We also filter all of our buffers with a 0.22 micron filter to ensure that there's no vesicles from other sources that may be present. And this is particularly important when we're reading, when we're using a particle counter such as the nanosite or Q-nano.
Now, Dr. Cheng, on your run report, what are the millions of reads that have no barcode?
Those are the reads that barcode were not detected, or the S5 torrent server was unable to determine, so it gets filtered out as a no barcode read and then placed into another file.
We have an audience member, John, who would like to know, why do you have bimodal read length and high average read length on the previous slide? Are you not filtering out larger sequences?
So in terms of the high average read length, so 55 base pairs, I forgot which slide that was on. So no, we're not filtering any larger sequences greater than the microRNA. So although I'm interested in microRNA, firstly I will map to microRNA or near base first. So that would be looking at the 22 nucleotide fragment lengths. And then what I do is with the other longer read lengths, such as 55 base pairs, etc., I will then map them to, for example, ensemble, and then determine whether any of those other longer read lengths are other types of small RNA biotypes, and that really carries into just other additional analysis to see whether we find, for example, tRNA or smallRNA, etc., for further investigative information.
John also is asking, what were the large number of no barcode samples? Have you performed additional analyses on those?
No, I haven't actually, but I believe these may be fragments whereby the barcode was not ligated into the fragments. So sometimes there will be, a certain percentage always, and whether this is high or low is dependent on the run itself.
Dr. Cheng, have you ever compared the miRNA profiles between serum and plasma?
Yes, so if you go back to the beginning of my presentation, I'll just see if I can do that. I did publish this work when I compared serum and plasma in GEV, if you want to look that up. So that slide here is serum and plasma. So this is looking at neat serum and plasma, where you can see the differences between the small RNA profiles. In terms of the small RNA profiles that I sequenced, they can be sometimes slightly different, and you can look that up in this paper, Cheng et al., GEV 2014.
Okay, how about, have you used other library preparation methods that employ adapters that decrease overall adapter dimers via chemical modifications, for example, TriLink?
No, I haven't tried it. Ideally, what we want to do is to obviously minimize primer adapters. Today I've shown you some of my tips. At the end, ideally, yes, we do want to develop a kit that will reduce primer dimers. Unfortunately, it's quite difficult when some of our samples will only have 1 nanogram of RNA available for us to make small RNA library.
Dr. Cheng, have you looked at miRNA presence within microvesicles as well? Is there a reason you have focused specifically on exosomes?
Yeah, good question. We have profiled small RNA from microvesicles as well as apoptotic bodies. And again, they produce a different small RNA profile where there is less microRNA in microvesicles and apoptotic bodies. So if you look at some of the papers that I've shown here, we have isolated and sequenced microvesicles in this paper published in RNA Biology, which I'll point to here, as well as in this year we also published another one more recently, again looking at microvesicles and apoptotic bodies. Depending on the cell line, the various vesicles will produce different percentages of small RNA species. So you can have a look at these publications and look at the breakdown of small RNA biotypes in various cell lines and vesicles as well.
Dr. Cheng, Jolene from the University of Calgary would like to know, is the process of PBS washing necessary in ultracentrifugation?
Yeah, it depends on what you want to do with your exosomes later. So I find that for some samples, performing the PBS wash, you may end up losing a lot more exosomes than gaining. If you want to perform functional assays with your exosome pellet, I think it is important to wash your exosomes with PBS and then spin them again. We find with the biomarker work, if there is any protein contamination, it will be removed during the RNA extraction. So the PBS wash isn't as important.
How much serum is required for miRNA analysis?
For NGS sequencing, if you are isolating exosomes from serum and plasma, I usually need a minimum of 1 mil. After the NGS process and you want to just do the validation by qPCR, I usually use anything between 300 to 500 microliters of serum and plasma, followed by the exosome extraction method.
Sorry about that. We have time for just a few more questions. Have you compared EV miRNA profile versus circulating miRNA profile in serum?
Yes, I have. I have compared both between the EV profiles and the neat profiles. Again, this was published in this paper here in JEV in 2014, where I compared the profiles from both serum and plasma, isolated from exosomes isolated by the ultracentrifuge, as well as a commercial kit from Norgen, and I compared these profiles to neat profiles from plasma and serum. So you can have a look at that profile as well, or that publication.
Thank you, Dr. Cheng. And we will close with this last question. What methods are there to isolate exosomes for diagnostic testing?
So depending on what you want to do, there's several methods. You can use the ultracentrifugation protocol, various commercial kits that are currently now on the market. There's many of them, and I suggest that you try them all to see whether in your hands you can isolate enough RNA from your serum and plasma volume, and that whether that amount works for library construction.
Thank you again, Dr. Cheng, and thank you for your important research. Do you have any final comments for our audience?
No, all the best with your sequencing project.
I would like to once again thank Dr. Cheng for her presentation, and I would also like to thank LabRoots and Thermo Fisher Scientific for making today's educational webcast possible. Now, before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thank you for joining us. We hope to see you again soon. Goodbye.